Welcome to Shalo's Biology. In today's lecture, we are going to study about primers and what are the criteria of selecting a primer. Then I'll teach you how you can design your own primers for the PCR. So let's begin. Primers are short single-stranded DNA sequences. They are complementary to the target sequence and they are consist of 18 to 30 nucleotides. Why you need primers when you have DNA polymerase? Because DNA polymerase is the enzyme which are actually going to synthesize your DNA. The reason is this enzyme cannot initiate the reaction by itself because it requires free hydroxyl group at the 3' end. It requires free hydroxyl group at the 3' end to initiate the reaction. It can continue the already existing strand by adding new nucleotides. So that's why we provide the primers in this reaction, and the primer, as it is complementary to your strand, it's gonna attach to the strand, and then you have a free hydroxyl group which DNA polymerase can use to add new nucleotides. So that's how DNA actually synthesizes with the help of primers, and this is why we provide primers in your PCR reaction. There are two types of primers, forward primer and reverse primer. As you know, the DNA is a double helix and it consists of two strands. When these two strands are separate, you have to multiply both of them. You have to synthesize both the strands in order to make identical copy of your original template. So that's why we designed two primers for each strand. Here we have two strands, so we design one primer for the upper strand and one primer for the lower strand. So forward and reverse. These two primers carried out the whole reaction. Next, what are the criteria of selecting your primer? They're based on the following factors. First of all, we have length. Length is very important because if your primer is too short, there is a chance of non-specific binding. What does it mean? That if your primer is too short, there is a chance that the similar sequence or complementary sequence might be present somewhere else in your DNA. Let's suppose you design your primer for this region, but your primer was too short, so there is a chance that the similar region, similar sequence might also be present here, so it result into non-specific binding. And if the length is too long, then there is a chance that your hybridization rate slows down. So in both way, your reaction will not give you your desired product. That's why the optimum length should be 18 to 22 nucleotide. This is the ideal condition. Next we have melting temperature. Melting temperature, or also known as annealing temperature. This is the temperature at which your primers are going to bind to your template. We have a formula to multiply by adenine plus thymine plus 4 multiplied by guanine plus cytosine. So by using this formula, you can easily find out the annealing temperature of your primer. It usually varies from 45 to 65 degrees. It can be any temperature depending upon your sequence. So here I have an example of a primer. In this sequence, I have total 20 nucleotides, and out of which I have 11 adenine plus thymine, and I have 9 guanine plus cytosine. Give me 58 degree. So the melting temperature of this primer is 58 degree. That's how you can also find out the TM temperature of your primers by using this formula. Next we have GC content. It is very important. GC content is low, then there is a chance that your primer will not bind, or if it is already binding, there is a chance that it lose and go away. Or if there is high GC content, then again there is a chance of slowdowning of your hybridization process. That's why the GC content must be 40 to 60%. That's the ideal case. Here we have a formula through which you can find out your GC content. GC, GC content, number of GC divided by total number of nucleotides multiplied by 100. In this case, as we have 9 guanine and cytosine divided by 20, total number of nucleotides multiplied by 100, and it will give us 45 degree. So in this case, I have 45% GC content. So GC content also promotes strong binding and specificity in your primers. Next we have secondary structure. There is a chance that your primers make secondary structure and it will hinder your PCR. How they can be made? Let's suppose you have a sequence A, T, C, G, T, A, G, A, T. This is a sequence and it has a complementary region. As you can see, ATC, GAT. If this primer folds itself in that way so that the GAT, the complementary strand, come in front of it and they start making bond. In this case, this is the secondary structure known as hairpin. If your primers has complementary sequences, there is a chance that they bind by themselves and make hairpin or secondary structures which will hinder your PCR. So you must avoid the secondary structures. Next we have the distance between the primers. What does mean the different distance between the primers? Basically, as you have two primers, forward primers and the reverse primers. Here, let's suppose you have this forward primer and this reverse primer. There is a distance between both of them, distance of nucleotides. If that distance is too much, if it is more than 10 kb, there is a chance of misbinding. There is a chance of misbinding. And if it's less than 150, then there is a chance of non-specific binding. So to avoid that, you must keep your distance between the primers 150 base pair to 10 kb. So these are the ideal condition of selecting a primer. You must keep in mind these factors while designing your primer. So now we are going to design primers. Let's suppose this is your gene of interest or your targeted sequence. As you know, DNA is double stranded. This is the first strand and this is your second strand. You need two primers, one forward primer and one the reverse primer. So how you're gonna design it? You need a complementary sequence. So we'll make a complementary sequence G. So this is the complementary sequence of this and it's from 5 prime to 3 prime direction. So we'll send this sequence to the company so that they make our primers according to our sequence. Next we have the reverse. We'll start making our primer from the reverse direction. So this primer is from 5 prime to 3 prime. Now what you're gonna do is you invert it into the 5 prime to 3' direction, 5' to 3' direction. So this is your reverse primer. That's how you can design your primers manually. But now we also have so many different bioinformatic tools to design these primers. You can also use them, or you can manually design them. Or if you have your primers, you can also check them by this method. It's time to check if you get it or not. Quiz time! So here I made 3 sequences, and you have to tell which one is the best one. So first of all, we'll check the length. 21. Okay. 3, 3, 6, 3, 9. 9. So this is a bad option. You cannot have that much short sequence because it can give you non-specific binding, as we have already discussed. Also 21. This means 3 and 1 can be considered, but the option 2 must be eliminated because this sequence is too short. Next, we'll see the GC content. The GC content of the first option is 28%, while for the third option, for this primer, we have 61.9%. So this one is the best option out of these two, because the GC content must be between 40 to 60% to give the desired product. I hope I answer all of your queries. If you like the video, please hit the bell button and subscribe my channels for more videos. And also to give me comments about the topics you want to study with me.